So we'll get started for this, the posters and the flash talks. So my name is Trevor Bushell, current BNA Treasurer. As I said, if you were at the AGM yesterday, I've only got 1 year to go. So if anybody's interested in taking over the role, feel free and I'll have a chat with you. But we've got these posters today, these flash posters, elevator pitch. It's a common thing these days, but I think it's a great way for people to sell their work, sell their students, what they're doing. Difference today from the last couple of days, we're not going to have questions. We've got 10 to get through, so we're not going to have questions, okay? So I think the last couple of days we had questions. We're not going to have questions, but obviously the whole point of this is to sell their posters so that you go and talk to them afterwards. So please go and see them. It's the last day. Let's support everybody who's doing the presenting on the last day as well. So up first, we've got Tim. We're sticking to 3 minutes. Should have been 2 slides. It's gone a little bit longer than that, but we're sticking to 3 minutes and we're sticking, so I'll be standing up and basically kicking them off at 3 minutes, okay? We're sticking to that, okay? So, Tim, please, on you go.
Hi, thanks very much for the opportunity to talk to you today about our work trying to look at the link between type 2 diabetes and Alzheimer's disease, which I'm sure you all know is the most common cause of dementia. Alzheimer's disease, 95% of it, about 95% is late onset or sporadic. And for this, we know there are various different risk factors. Probably the 2 most important are age and the ApoE3 common allelic variant. We don't know exactly how sporadic Alzheimer's disease starts, but we do know that you get the appearance of these amyloid beta plaques and tau tangles, and that both age and ApoE4 increase the risk of you developing these. But more recently, it's been recognized another potential hallmark of Alzheimer's disease is the inhibition of adult hippocampal neurogenesis. And this has been shown in both human patients and animal models. And actually, interestingly, it's also shown in animal models of type 2 diabetes, which is another major risk factor for late onset Alzheimer's, increasing the risk by about 75%. We don't understand why, but our hypothesis in our lab is that the various different factors that change in type 2 diabetes, and the one we're focusing on is free fatty acids, inhibit adult hippocampal neurogenesis. And it's been known for a while that fatty acids can be neurotoxic, but these generally are applied at very high levels, which recent research has suggested don't represent the environment in the brain in type 2 diabetes. So we've tried this with low chronic exposure to free fatty acids that we think is more disease relevant. So what have we done here? Well, we've used a cell model for looking at neurogenesis. We looked at the differentiation of SHSY5Y cells, which seem to follow most of the pathways between neuronal precursor cells and immature neurons. And using BDNF and retinoic acid, we can differentiate these cells from something that looks fairly precursor-like into something that looks decidedly neuronal within 10 days. And we see when we introduce free fatty acids into the system at the level that's been shown to exist in the brain of people with type 2 diabetes, that we get a severe inhibition or dysfunction in this neurogenesis and this neuronal differentiation, including a reduction in synaptic markers such as synaptophysin and PSD95, but also a disruption of key signaling pathways like AKT and GSK3 beta, and these have also been shown to be intimately involved in the pathology of Alzheimer's disease. There's loads more data on my poster. Please come and talk to me about it. But we think, in summary, that free fatty acid exposure during differentiation of neurons is one of the key links between type 2 diabetes and Alzheimer's disease. Thank you very much.
Thanks, Tim. Much appreciated. Great start there. Good timing. Perfect timing. Obviously, so something to live up to for the rest of the presenters now, of keeping that time. So up next, we have Chloe. And Chloe's talking about inter-individual variability in development trajectories of brain volume across adolescence. And she's going to tell you more. Thank you.
Hi everyone, my name is Chloe. I am a PhD student at King's College London, and today I'm presenting some findings from my PhD work about individual differences in developmental trajectories of brain volume across adolescence. So, as a teenager, your brain undergoes a period of immense change, and that includes decreases in cortical volume, increases in white matter volume, decreases in total brain volume, and a varied pattern of volumetric increases and decreases in subcortical volumes. And while research has done a very good job at differentiating what's happening on average in these different brain structures, we know less about how individuals differ in their developmental trajectories. So the aim of my study was twofold. Firstly, to examine precisely the shape of development in these different brain regions, and then also to quantify individual differences in these developmental trajectories. I used a sample of Danish participants, so there were 90 participants aged between 7 and 21 years, and there were up to 12 repeated MRI measurements per individual across that time range, so that's a very unique data set. So firstly, to answer the question of what the shape of development is in different brain regions, I used an analysis approach called generalized additive mixed models. So this is a data-driven, bottom-up approach that allows us to flexibly capture change in different brain regions. So you can see here for global brain regions, with age on the x-axis, volume on the y-axis. In each brain region, there are distinct patterns of volumetric development that were followed. So we present the average trajectory with the dotted black line, the average female trajectory is in purple, and the average male trajectory is in green. And the same with subcortical regions. So these findings show us that in each brain region, there are distinct patterns of development followed. To answer the question to how individuals differ in developmental trajectories, we used another analysis approach called nonlinear mixed modeling. I'll give an example for the cortex. I have many different findings for the different brain structures, so please come to my poster afterwards. But for example, in the cortex, we found that an S-shaped logistic model was the best capture of volumetric development in this region. For each individual, using this nonlinear mixed modeling framework, we were able to extract the inflection point. This inflection point corresponds with the age at which volume is changing most rapidly in that sample. So for each individual across the sample, you can see that there's a vast range of inflection points. So some people had their volumetric development much earlier in development, and then others much later. And there were also sex differences in those individual differences in the volumetric trajectories. So key takeaways to wrap it all up. First of all, this study is one of the first to show that individuals differ not only in the pace of volumetric development, but at the age at which they're reaching key developmental milestones. And brain regions follow very distinct patterns of development across adolescence. Thank you very much.
Whoa, I was just about to come in. Well done. Thank you, Chloe. Thanks very much. As I've said, if you've got any questions, please go see their posters and go and have some interesting ones. And the male-female differences there is always very interesting. So up next, we have Aileen. And he's using EEG to predict dementia risk and cognitive resilience in elderly patients undergoing surgery. Thank you very much.
Hello, I'm Aileen. I'm a clinical fellow at the University of Birmingham studying dynamic resilience.
The wrong slides. Yeah, it should be Aileen. Yeah. Okay. No problems. Rather than for you having to carry on talking. Right, there we go. Right, thank you. Okay.
Start again. So I'm Aileen. I'm a clinical fellow at the University of Birmingham studying dynamic resilience in older adults undergoing surgery. Older adults often experience cognitive decline following surgery, which commonly presents as delirium, delirium being an acute disturbance in consciousness and attention. Changes in attention control networks can be detected through the attention network task, which is seen here. In this task, participants are presented with 5 arrows, and they're told to respond to the direction of the middle arrow. The flanking arrows can be either congruent, facing in the same direction as the middle arrow, or incongruent, which is facing in the opposite direction of the middle arrow. This is the executive control part of the attention networks. Preceding the target, there may be cues that can alert participants to attend this is alerting, or even direct their attention to the spatial location of the target, and this is orienting. Using a 32 electrode EEG cap, we'll measure brain oscillations time-locked to specific stimuli during the ANT, enabling real-time measures of changes in brain neuronal activity during responses requiring activation of the alerting, orienting, and executive function networks. As we age, cells change, which is called senescence, and they release inflammatory mediators into the blood known as SASP. This chronic low-grade aging-related inflammation is termed inflammaging. In a previous study seen here, we've shown that inflammation alters brain oscillatory activity, specifically in alpha waves, during the ANT task. Combining biological measures of inflammation with EEG measures has potential for development of biomarkers predicting risk of post-operative cognitive decline. And this is particularly important for older adults undergoing surgery as they're at higher risk of developing cognitive changes post-operatively. In 86 over 65-year-olds scheduled for surgery, we will take clinical measures of physical and cognitive function along with blood samples before and after surgery. Importantly, we'll also record EEG, neural oscillatory activity, while patients perform the ANT task to further understand the attentional networks before and after surgery. We will assess the presence of delirium within the 3 days immediately following surgery to find those whose brains are least resilient. We hypothesise that higher preoperative markers of biological inflammation and a greater alpha-power suppression during alerting when tested preoperatively will be observed in those presenting with post-operative cognitive decline when compared to those who demonstrate cognitive resilience. The alpha-power modulation is likely to reflect the greater resources required to maintain cognitive function in those most susceptible to decline. Thank you for listening, and I'm happy to answer any questions over at my post afterwards.
Brilliant. Good timing again. All good examples, all been practicing, so yeah, don't go over time. I've not had to kick anybody off yet. So up next, we've got Z. Yep. And Z is going to be introducing post-surgical tactile evoked pain, a role for BDNF track B dependent novel tactile corpuscles.
Hi, I'm Z. I'm from the Torsney Lab from the University of Edinburgh, and we study responses to touch and pain. So millions of patients undergo surgery each year, but a significant proportion of them develop post-surgical pain. So one of the most common and disturbing symptoms is tactile evoked pain, where gentle innocuous stimuli such as the brushing of clothes against the skin surface induces pain. So what we're trying to do here is look at the peripheral mechanisms that may contribute to this. So to do this, we use the hind paw incision model in the rat, where an incision is made in the heel, in the skin and the underlying muscle, and it is sutured and glued. So with this, we see tactile evoked pain behavior on the brush assay, and this peaks at post-surgical day 2, and it resolves by post-surgical day 7. So in order to study the peripheral mechanisms, we looked at skin structure and innervation. And using collagen 4, we saw that there are these dermal protrusions that are oriented towards the incision site. So usually, these dermal papillae are not present in the heel, but they are present in a rat foot pad. So usually in the rat foot pad, these dermal papillae contains the tactile corpuscles, which consists of a neurofilament nerve fiber associated with S100. And we wanted to see if these putative dermal papillae also contains these tactile corpuscles. So indeed, in these two pictures, we can see that these dermal papillae in the incision heel do indeed contain these tactile corpuscles, and when we quantified them, they are expressed in a pattern that matches the time course of the pain behavior. And next, we wanted to look at the role of BDNF track B in this, as we know that BDNF track B signaling is involved in their development, and also we found that BDNF intensity is increased in the skin around incision site. So to do this, we injected intraplantar TREC-BFC on day 0 and day 2 after the surgery, and we did pain behavior testing on post-surgical day 3. So here we see that in the TREC-BFC injected group, they show a decrease in tactile evoked pain as compared to the incision-only group. And when we quantified these structures, we did not see a difference in the density of these structures. However, the S100 bulb area was decreased, so it suggests that these tactile corpuscles are smaller. So in conclusion, in the incision sites around, we see the development of these novel tactile corpuscles that develop in a BDNF track B manner, and we suggest that this provides the tactile drive that induces tactile evoked pain. Thank you.
Perfect timing again. Thank you very much indeed. Really interesting stuff. So up next, we have Fatima. Thanks, Fatima. So Fatima's Federated Data Analytics, so case example of an international collaboration for cross-institutional data sharing for dementia research. On you go, Fatima.
Thank you very much. Thanks for having me. I'm going to flip the theme a little bit. So I'm Fatima Torabi. I'm a senior research data scientist at Dementias Platform UK, and I'm also an assistant professor of healthcare data at the University of Cambridge. What I'm going to talk to you is about federated analytics. So all of the studies we've observed and presented before me have gathered some data. If these data are patient data, we need to keep them safe. We need to ensure that the required governance are in place. And that's where we see the development of what we call trusted research environments. So in these environments, we are trying to adhere to 5 safes. Mainly, we only give access to safe people. All the projects go through the safety checks to ensure they are developed to the benefit of our population and patients. The data is safe. Mainly, it's anonymized before getting deposited into these environments. The setting is safe. To give you an example, you're disconnected from internet when you're working in these environments, and a lot of other safety measures in terms of computational establishment. And also, any output coming out of these environments go through a check, disclosure control check, to ensure that we can't access the patient level data. So this means those data often held in a silo. So if it's deposited in a single environment, then it has to stay there within the governance of that environment. So my use case when I came to lead this project was I had a handful of cases with specific conditions, and I needed to access their data from across different trusted research environments. So how could I bring these data together? How could I achieve this? And the use case brought me to see if we can establish an ecosystem of trusted research environments who are able to share data across themselves. So I wrote a commentary and you have the link to read it. This is establishing a governance model that allowed us to spark in this journey. And then the next one, we actually were able to achieve it between 2 environments in an international setting. So the data held in Dementia's platform UK were accessed from ADDI platform, meaning the researchers can develop their analytical pipeline. I was glad to see Chloe and all the colleagues before me establish an analytical pipeline, a structured protocol to hit those data sets. So it is possible, and we are able to facilitate those environments. I'm out of time. I'm going to be the good one kicked out. Come talk to me to poster.
Thank you very much, Fatima. Thank you. Thank you indeed. So up next, we have Rebecca. And Alzheimer's disease models in iPSC-derived glutamatergic neurons show increased secretion of pathogenic amyloid beta peptides.
Doesn't show anything.
Yeah.
Hi everyone, thank you for this opportunity to present our work. My name is Rebecca Northeast and I'm a Senior Product Manager at BitBio. So BitBio is a synthetic biology company spun out from Cambridge University, and we make iPSC-derived cells using our forward programming technology, which we call Optiox. So using Optiox, we're able to generate things like our glutamatergic neurons, which are an excellent model to study Alzheimer's disease. Oh, sorry, that video is meant to play. But what the video would show is that these neurons develop rapidly, and you can see they're assay ready in just 11 days following this 2-phase protocol, and they develop visible neural networks by day 5. Using our glutamatergic neuron wild-type iPSC line, we were then able to engineer Alzheimer's disease mutations in presenilin-1, APP London, and APP Swedish mutations. And these act as an in vitro model to study Alzheimer's disease. So what we wanted to show, or to see, is to assess whether or not you could see a similar phenotype that you would expect to see in amyloid beta peptide secretion that you would see in patients. So to do this, we cultured our cells up to day 30, and we measured the amyloid beta peptides 30, 38, 40, and 42 using a multiplex ELISA. And then in our presenilin-1 mutations, we see that you have an increase in A beta 42, which leads to a significant increase in the A beta 42 and 38 ratio and the A beta 42 and 40 ratio. In our APP London mutations, we see a significant increase in the amyloid beta 42 and 38, but not in the amyloid beta 40 peptides, and this therefore leads to a significant increase in just the A beta 42 and 40 ratio. And then finally, in our APP Swedish mutation neurons, we see a significant increase in all the peptides, so in the A beta 38, 40, and 42, So this therefore leads to no change in the amyloid beta ratios. And what you can really nicely see here is that this is the expected phenotype of the A beta peptide that you would expect to see across these mutations. And also in the homozygous and heterozygous mutations, in the homozygous you can see there is a higher secretion of peptides that you would, again, which you would expect to see. So in conclusion, these cells act as a really great model to study Alzheimer's disease in an in vitro way, but also they're essentially used by pharma companies to look at screening compounds that might modulate amyloid beta secretion. And the way that these neurons are made also means that they are incredibly lot-to-lot consistent, so they provide a really functional and consistent model to study. If you're interested in finding out more either about these cells or about BitBio, please come and see me at my poster number 6. It's opposite the Hello Bio stand. Thank you for your time.
Thank you very much, Rebecca. Again, keeping to time, which is great. Making my job easy, everybody, which is nice. Okay, so up next we have Vasilis. And Vasilis is going to be using functional connectivity to validate individual specific localization of the auditory thalamus. Thanks, Vasilis.
Yep.
Whenever you're ready.
Hi, thank you. So my poster is number 147, entitled Using Functional Connectivity to Validate Individual-Specific Localization of the Auditory Thalamus. So the auditory part of the thalamus, or MGB for short, is important because it is one of the main subcortical relays of the auditory signal before the signal reaches the auditory cortex for further processing. So given how centrally positioned it is in the auditory pathway, it's not surprising that the MGB is implicated in hearing functions and disorders like tinnitus. However, precise identification and systematic measurement of the MGB in humans is very challenging, partly because it is located deeply, it has a small size and variable anatomy across individuals. So here we used UK Biobank data to localize the MGB in every individual based on its anatomical connectivity to the primary auditory cortex. To do that, we basically used MRI-based tractography to trace tracts between every voxel in the thalamus and some specific target cortical locations that correspond to known thalamic connection sites. And to get the MGB, we basically classified the voxels based on their connectivity to the primary auditory cortex with higher probability compared to the other cortical regions. Then we estimated functional connectivity between the MGB and the cortex in order to validate our localization approach. And we found statistically significant, stronger functional connectivity between the cortex and our subject-specific tailored MGB compared to a situation where the MGB was taken from an atlas, from Jülich atlas, meaning that it is not subject-specific, it's a population average, indicating, that this approach is probably advantageous for the localization to be functionally relevant. Finally, we also looked at functional connectivity between the MGB and several other regions of interest that relate to tinnitus, looking at group differences between tinnitus and no tinnitus controls. We have found some interesting results here, but to find out more, please visit my poster. Thank you.
Thank you, Vasilis. Once again, perfect timing. Good to see people have been practicing. Up next, we have Monica, and her title is Thalamic Place Signal Driven by Subiculum?
Yes. So, hello everybody, I'm Monica. I'm a PhD student in Kate Jeffries' lab in Glasgow. And yeah, I'm going to present a project that I'm hoping to do soon. So, spatial navigation is a product of neuronal activity and interactions within the memory circuit, which is comprised by the hippocampal formation, prefrontal and retrosplenial cortices, and middle and thalamic nuclei. Now, within all of those regions, there are spatially modulated neurons, such as place cells, which fire when the animal is in a specific location within their environment. Now, place cells have been first reported in the 70s from the hippocampus, and they have been studied extensively since. However, recently they have also been found in the thalamus, specifically in the anterior thalamic nuclei, ATN, and in nucleus reunions. So this discovery suggests that there must be some sort of hierarchical organization of place signaling across multiple brain structures. However, what we don't know is what regions actually dictate place cell activity in the thalamus. In other words, where does the place signal come from in the thalamus? So here we hypothesize that it is the subiculum that drives place cell activity in the ATN, subiculum being a major hippocampal output structure that also is characterized by spatially modulated neurons, including place cells. So in this project, we're going to use chemogenetics in a mouse model to inhibit this projection and see what effects that has on the place cell activity in the thalamus. So just briefly, we're going to use a Cre-LoxP approach to make it very specific. So a Cre-dependent chemogenetic tool is going to be delivered to dorsal subiculum, and a Cre recombinase, retrograde Cre recombinase, is going to be delivered to ATN, from which it will travel to dorsal subiculum. This will allow for the expression of chemogenetic tool specifically in those neurons that project to ATN, as opposed to the whole neuronal population in the subiculum. We will also implant some tetrodes in the ATN, and then we will collect place cell recordings from the ATN from mice, freely moving mice during open field exploration. The mice will then be given chemogenetic agonist, which will again inhibit our projection, and then we will record those cells, the same cell as pre-inhibition again. What we expect to see is disruption of place cell activity. This will be evident as widening, broadening of the place fields and changes in place firing characteristics such as spiking frequency, spiking properties, and so on. Overall, this will give us a bit more information about how the ATN and the subiculum contribute to place representation in the brain, and the mechanisms that underlie spatial navigation. Thank you for your attention, and please come check out my poster. Thank you.
Thanks, Monica. Much appreciated. Keeping to time as well, right in point. Okay. Second to last, so we have Meg now, who is talk is on, our poster is about amygdala burnout, altered trajectory of amygdala volume associated with early life stress. When you're ready.
Okay. Hello, I'm here to talk about altered trajectories of grey matter volume in the amygdala associated with early life stress. So early life stress is classified as one significant or several bouts of cumulative stress that occurs before the age of 18, and it's generally categorized into abuse, neglect, and general household dysfunction. One region of the brain that's associated with early life stress throughout the literature is the amygdala. So the amygdala is a small almond-shaped structure that consists of multiple different heterogeneous nuclei and is associated with fear processing and fear learning. What's really interesting is that throughout the literature, there's no consistency in terms of the grey matter volume change in the amygdala, with some research indicating there's an increase and others indicating there's a decrease. A recent meta-analysis suggested that the age at which an individual is scanned may actually be explaining this discrepancy, with younger participants tending to show increases in volume and older participants tending to show decreases in volume. So we aim to test this. So first, for our juvenile sample, we conducted a rodent study where we induced a chronic immobilization stressor multiple times a week and imaged at 3 different time points. We used a deformation-based morphometry approach to estimate the volume change within the amygdala, and we identified that comparing the time points after stress to control, there was a significant increase in one of the key nuclei of the amygdala. We then looked at an older adult sample. So in order to do this, we used the UK Biobank, which is a large-scale biomedical resource. So in order to characterize early life stress, we used a shortened version of the childhood trauma questionnaire, and we used pre-processed limbic IDPs, which are essentially just volumetric metrics of the limbic system. We had a large sample of almost 25,500 people, and we conducted some straightforward linear regression models controlling for age and sex. We found that there was a significant reduction in the grey matter volume of the amygdala in our older adult sample. We then replicated this on a smaller subset of this sample. So we took those with the most extreme instances of early life stress and compared them against those with no instances of early life stress. We used the structural MRI scans from the Biobank database, and we did a voxel-wise region of interest approach. And we again identified, irrespective of the methodology, that there were significant reductions in the grey matter volume of the amygdala. So what this tells us is that in the short term, there appears to be these volumetric increases in the amygdala, whereas in the long term, in older adults, there appears to be this decrease, which could potentially suggest an altered trajectory of development for those who've gone through early life stress. So our next stage is to really identify the shape of this curve. At what point does the amygdala stop increasing in volume and start decreasing in volume, which we've titled point of burnout. So in order to do this, we're going to be using some other large-scale databases of young adults and adolescents, including the Imogen and ABCD dataset, and an additional rat study. Thank you to everyone, and thank you for listening. Thank you.
Thanks, much appreciated. So last but not least, we have Yuping Yang, whose talk is Thalamic Network Controllability Predicts Cognitive Impairment in Multiple Sclerosis. Whenever you're ready.
Thank you. Hi, I'm Yuping. My study focuses on understanding the state transitions in cognitive impairments in multiple sclerosis. So for human brain, the brain needs to transit between different states to get ready for different cognitive tasks. And in individuals with multiple sclerosis, especially those who develop cognitive impairments, the transitions become less frequently and require more energy. So in our study, we aim to test which brain regions are responsible for those changes in state transitions and how can this relate to cognitive impairments. So to do this, we applied network control theory to quantify the ability of regions driving state transitions in 2 dimensions. One dimension is average controllability, which quantify the ability to drive those low energy transitions, which are important for those simple tasks, like when you say hello to someone. The other dimension is model controllability, which quantifies the ability to drive those high energy state transitions, which are more important to those complex cognitive functions. And we found in those individuals with multiple sclerosis, especially those who developed cognitive impairments, the telomeres shows more increased average controllability and decreased model controllability. This suggests that the telomeres in this condition tend to drive the brain to perform more those low energy transitions and perform fewer high energy transitions. And given that telomeres is the most vulnerable regions in the multiple sclerosis, we interpret this as a energy saving mode, that the telomeres try to save energy to maintain those basic fundamentals neural activities, and as a consequence, the energy is not sufficient enough to support for those high energy cognitive tasks. That helps explain why they developed cognitive impairments in those patients. This figure shows the performance of predict models based on thalamic controllability in this study, and also based on thalamic volume. And our results found thalamic controllability models perform better than thalamic volume in differentiating multiple sclerosis individuals from health controls, and also in predicting cognitive performance in this condition. Thanks.
Thank you very much. Brilliant. So that was the last of those. So once again, can we have another round of applause for all the post presenters? Definitely get a round of applause from me for keeping to time and not make me have to be a bouncer and kick you off the stage. Please also go support final afternoon of the conference. Please go support the poster presenters on this final afternoon. Go talk to them, go chat to them. Even if it's not in your own area, go talk and chat to them. You always have some interesting points come across from there. But thank you, everybody. Thank you for coming, and hope you enjoy the rest of your day.